Speaker is going to be Enes Afghan, talking about enabling compute clusters on top of OpenStack. Tristan's been kind enough to sort of pass on some introductions to myself. Enes is a research scientist at the Centre for Informatics and Computing at Ruda Boschevich Institute, and is affiliated with the Taylor Lab at Emory University and the Galaxy Project. As he's setting up here, can I also very briefly mention that it is quite important that we don't bring food or drinks into the auditorium, and if you do, so keep them well and truly sealed. How are you going there? Enes, fire away. Thank you very much, Enes.
All right, excellent. Hi everyone. As was just mentioned, so this project is sort of continuing. So the name of the project that I'm going to spend most of my time talking about is called Cloudman, and I started that as part of my postdoc at as part of the Galaxy project and at the Emory University, and it's continuing now to be developed by both the Galaxy project and the Genomics Virtual Lab project, which I'll mention a bit later as well, and is one of the things that Tom mentioned this morning. So sort of the idea and the premise behind this this project was that, well, several times today it was mentioned that, especially from the end user's standpoint and sort of the researcher's standpoint, that they're primarily interested in sort of having a ready-to-use service. So something that ideally they just go pull up in their browser and start doing things. And at the same time, the cloud was sort of emerging, and it was quite obvious that it's pretty cool technology that makes the compute capacity available and the compute resources available to those researchers. But the infrastructure component of it is a bit problematic in that you really don't want to have to, or the researchers per se don't want to deal with individual machines, with hard disks, with file systems and so on. Instead, they're more interested in this software layer of the component. And so that's where the idea of Cloudman emerged, as sort of this bridge as a sort of a lightweight platform as well that bridges the gap between the flexibility of the infrastructure with the functionality of the software as a service. And so the idea is to take individual resources, individual components, group them into meaningful units and expose that via either a web browser or a standard interface that researchers are already used to. So that's where sort of Cloudman comes in, and what it does is it's basically an orchestrator. It orchestrates all the steps required to provision, manage, and share compute platforms on a cloud infrastructure, and primarily through a web browser. So more specifically, it allows one to create a dynamically scalable compute cluster on top of otherwise just disparate instances, and then comes pre-configured with several bioinformatics applications, because that was the domain in which it was originally created, but you can add your own, whether they be in bioinformatics domain or any other, and then once it's all done, including the data that might have gotten uploaded, you can share it with the world or with other users. And so the process of deploying one is basically this list of 3 steps. So you need an account on a cloud, and you start the master instance just like you would on a cluster. You log into a master node. Well, here you're launching your own master node, and then you get the Cloudman interface in a browser that allows you then to manage that cluster and the services that are running on that cluster. So starting one up is when there's 2 methods. You can either use the native cloud dashboard, or you can use this web form that we've created as a way to sort of get going. And so I'm going to sort of jump in and do this in parallel. I didn't want to do just the demo because it was going to leave me in this really questionable position of either having not too much to talk about, and things happening too fast, or having too much to talk about. So we'll launch one, and then I'll go back to the PowerPoint and talk and as things happen, because each of these steps takes a couple of minutes. So each cluster requires a name. So we'll give it a name. We'll give it a password that protects our web portal accessing, and then we choose which cloud we want to run on. We'll choose the Nectar OpenStack cloud, since that's the topic of this. We put in our SSH keys, which are conveniently here. We choose the instance type, choose something a bit larger, and we launch one. And so this will take a couple of minutes, and so I'll just so once this happens, we'll get a URL and all. But in the meantime, we'll just go back to the PowerPoint. All right. Yeah. So once it launches, we'll get the CloudMint front end, the web interface. You have 4 types of clusters that you can launch, basically. So the default one being a Galaxy cluster. I'll talk more about Galaxy in a minute, but we'll just keep that in mind. The other one is the other option is a shared instance of a cluster. So what I mentioned, you can instantiate somebody else's shared instance. I'll talk more about that later as well. You can share a data cluster. I mean, you can start a data cluster, meaning you have a persistent data repository, but no applications other than what's installed on the base image. But it's persistent, meaning you can terminate it. You can come back, use the same cluster name, launch it, and all your data will be there. And then lastly is just a sort of a test cluster, meaning you launch one, but once you've terminated it, it's all gone. So it's useful if you have an analysis that you kind of want to do. And it just requires some compute, some capacity. You'll upload some data, some scripts that might already be running on a dedicated SG cluster you have at home or in your institution. And when it's all when the computation's done, you'll pull it back off. You'll pull the data back off the cluster, terminate, and the thing will disappear as if it never existed. Let's see if this thing launched on the not yet. All right, so once it does, once you choose your type, you get the interface up. It's basically divided into three sections. You have at the top, you have the control, so scaling primarily, adding and removing nodes. In the middle pane, so the center of the page is the status, cluster size, disk file system size, cluster name, the ability to share and things like that. And then you have the node view, the kind of the view of the nodes that are available in the cluster. So they're color-coded. Green means all good, yellow means something's happening, it's either being added or removed. And then the gray glyphs in the middle sort of indicate the load over the last 15 minutes on that particular node. And so as you scale up or down, that changes. And then the bottom bar, you can expand that into a log. Sort of see the system log of what's happening on the cluster at any point in time. I'll go back again. Well, still not there. All right. So once all the applications, so that basically sets you up. And at this point, the cluster is ready to be used. If it was a Galaxy type cluster, you can click on the Access Galaxy button, and you'll get Galaxy application up and running. And so what is Galaxy? I guess I'll digress a bit here and talk about Galaxy as a whole. So has any of you, or who has heard of the Galaxy project in the bioinformatics domain? Oh, wow. All right, so it's an open source web-based platform for performing accessible, reproducible, and transparent genomic science. What that sort of entails is that it's basically a web execution, a web-based job execution framework. So you can take any Unix command line tool, write a little XML-based wrapper script that defines it, and it'll expose it in a web front end. So that's the first premise of the Galaxy project. It's accessible, so it means it makes those tools accessible to researchers, to bioinformaticians, biologists. Secondly, it's reproducible, meaning that each step of the analysis, which sometimes can contain, oftentimes tens, if not hundreds, of steps in a given analysis, and each step is sort of monitored as, not monitored, but the parameters of each step are kept, so you can always go back and rerun or inspect what parameters you use. And some of these tools will take 5, 10, 15 different parameters with a large number of variation. You can always go back and check what parameters you used and rerun it. Similarly, or not similarly, but continuing in that space is that everything that is created within Galaxy is transparent, meaning that you can share it with either individuals or the world, thus making it public. And so other users, especially after you have published your research and your paper has been published, you share your components, the computational components that were done within Galaxy, and then users can continue and build on the results that you've already obtained, or make sure that the results were actually accurate in the way you describe them. And so Galaxy by itself can be just used right away as a free public service online. Just go to usegalaxy.org and you'll get an instance of Galaxy up and running. However, or an alternative to using the public one is to use to download the source, install it yourself, and what you get by that is the ability to customize it. So there's a predefined set of tools that's available on the main instance. There is a limited capacity in terms of resources, both compute and storage, that's actually made available to users. So if you download it and install everything by yourself, well, you remove those limitations. However, it requires you also to install and maintain all these things. And a lot of times we're talking 100+ bioinformatics tools. We're talking terabytes of reference data sets that are needed by these bioinformatics tools. And so there's this sort of trade-off of well, it's ready and convenient, but it's kind of hard and flexible. And so that's where the cloud sort of comes in and fills the gap of largely you get a functional and ready infrastructure with Galaxy, but without having to install all this stuff by yourself. And so one more slide on Galaxy, I guess. So, well, like I said, any Unix command line tool, whether it be bioinformatics or not, can be integrated into Galaxy. Means you just take a editor, write up an XML document, an XML file that describes what are the options for a given tool and how to invoke it. Once it's done, add it into the config file for Galaxy, and it'll just be listed as an additional tool in Galaxy in the web GUI. You choose the tool that you've just defined. It automatically creates the web front end for it. You specify the parameters as you wish. Submit run, and the job runs, whether that be on your local laptop or on the cluster. Galaxy handles that transparent or sort of automatically behind the scenes. And this you get an element in your analysis step, so an element in what's otherwise called a history, and you can now inspect, or you can first see, get your results and continue, or you can go back and inspect those parameters that I mentioned earlier that Galaxy keeps track of. And lastly, there's a visualization component to Galaxy called Trackster, built on HTML5 and very flexible, very scalable, that allows you to visualize data sets and things that can be visualized. So why is all this actually sort of necessary and all? And it's basically a problem with the size of data and the amount of data that's being produced today. So next generation sequencers are sort of in blunt terms maybe an X-ray of the future, where a lot of these are sort of being bought and started to being used around the world, and they produce enormous amounts of data. And the prediction is that they will just produce more and more data as as time passes on. And so the problem is that there's basically not enough bioinformaticians to be able to process all this data. And so the idea is to empower biologists themselves to via simple tools like we just saw to actually analyze the data that's coming off the machines and that's in the interest of their experiment and research. And so that's where the Genomics Virtual Lab comes in. So this is an Australian national project funded by Nectar and co-funded by the partnering institutions that basically tries to build an infrastructure in terms of both the compute and the capacity, and then going up to community resources, including tutorials, workshops, and best practices to enable both bioinformaticians and biologists to perform data analysis. So it's all underlined by this clustering the cloud component by Cloudman, then Cloud by Linux as a set of bioinformatics tools that's exposed. Galaxy on top of that as a tool in as a web front end to a large number of these tools, and then sort of in the community space create a sort of a number of tutorials, a number of workshops that teach people how to use this stuff, how to actually do bioinformatics and what these tools do, and then parallel to that create a set of best practice workflows or protocols that can actually be applied with some tweaking to actual research problems that people are encountering. And so I go back to talk more about the sort of cloud and cluster in the cloud concept and the cloud. And so, like I said, it's been under development for about 2.5, 3 years or so, it continues to be developed. So we've piled up several features that seem to be quite useful. So basically, with that web form and launching an instance or two, let's see what's actually happening there. Well, I don't know, something screwed up because it shouldn't take this long. Well, you can try it on your own. All right, so you basically get an SGE cluster, head node, and you can add workers as necessary. We've just added support for Hadoop-based clusters, which those are integrated into SGE, and then we're working on Condor, integrating with Condor and going with a sort of federated cluster approach. Well, you get the configure Galaxy. I'll kind of have to skip through this. So OpenStack support originated on Amazon. It's been ported to OpenStack, Eucalyptus, and OpenNebula. So basically any cloud middleware that exists today, you can get this working on. The process of adding it to a machine image is automated. So you run a script. It installs the dependencies, the packages, and you can basically add the cluster on the cloud compute capacity to any Ubuntu-based image at the moment. And so I see I'm running out of time, so I'm going to skip through some of these. Okay, and well, I'll just skip through all of them, and we'll talk about the we'll talk about a couple of features a bit later on. So skip through this one as well, and so to say that the idea is to really enable sort of this moving and reproducibility of user's environment. So you can go ahead and think that if you just look at a general or at a number of these platform as a service offerings, whether they be at the bioinformatics end or at the sort of just platform as a service end, oftentimes there's a question of lock-in and all that. And here the idea is to sort of bundle this stuff into a more traditional environment. So cluster compute environment with SGE is a pretty standard environment to run parallel workloads. And so you can sort of think that, okay, well now if I have that built for me automatically and I already have scripts that otherwise run on a cluster, but that cluster is full, I just copy things over and start using it. And then so if there's not enough capacity on this cloud, I'll go to another cloud, and ideally that will one day happen automatically. It makes it possible to sort of move because we're working with low enough components at both cloud level and at the sort of user interaction level to where the moving of things seems to be quite portable. So that's kind of the premise. And so on top of this set of features that are otherwise just available by either wrapping low-level cloud components or applications that otherwise already exist, there's a few features in CloudMan that are just value added and custom to its or a result of its own development. And so one is you can customize your instance. So each instance is completely self-contained, meaning that you can modify it as much as you'd like to. So if you want to add your own data, if you want to add your own tools, if you want to rip things out, if you want to use your own image, it's perfectly fine. And so it's kind of nice, I guess, that you're able to start off not from 0, but from like level 5 or so, and then just build on top of that. So for example, in bioinformatics, there's 100 tools that are already available by default, but not that 101 that you really need. Well, but that one requires 3 others, and so you don't actually have to install those 3 others and go through the process of configuring those. Instead, you just add the one that's missing, and so it gives you a head start. Next thing that I mentioned, because they are self-contained and some extra work went in, you can share that entire instance. So, for example, you have a tool, you have an application that you want to deploy, but it has a README file that's this long and requires a large number of configurations and whatever. Well, instead of having users go through that README file and install the thing themselves, you can install it yourself by customizing an instance and just share it. and either make it public or share it with an individual user. They derive a derived cluster from their shared instance and get a replica, an exact config of what you have performed. So instead of just being an execution platform, it can be a tool distribution platform as well. And then lastly, the auto-scaling thing, well, some of it natively comes from the cloud that you can dynamically scale up and down. So you just you choose the instance type you want to run, add it, but it also has this notion of auto-scaling, and it's not based on just on the CPU load, but instead on the size of the queue in the job manager. So if the queue is full and moving relatively slowly, it'll add more nodes, and as nodes sort of just start sitting idle, it'll kill them off and move things around. And terminate things and all. And so. Lastly, one architecture slide. Like I said, it's deployable on any Ubuntu-based image. It has a few dependencies on Ubuntu and you can just add it on. It has it keeps state in a persistent data repository, whether that be Swift or S3, and then it creates file systems based off of either volumes or snapshots that can be again shared or not shared. And we're currently in the process of adding. Basically an arbitrary data source to it as a file system. The first available feature is adding an S3 bucket as a local file system, and then we're soon going to have support for like an already existing NFS file system, or we'll eventually go into more distributed file systems type stuff, especially since I've been hearing about it a lot over the past 2 days. So anyway, I'm going to stop right there and
thank you very much, Enes. There we go. A very good overview.